everyone. In this video, we will be seeing the basic things that we must be knowing in bioinorganic chemistry, that is metalloporphyrin. Why is it important means I think the most important molecules we will be studying in bioinorganic chemistry contains this metalloporphyrin. So generally in bioinorganic chemistry, we'll be seeing the naturally occurring inorganic elements in various species. What is their structure, their structural positions, their action, what are their functions, everything. Then we also study about the drug action, like how drug reacts with our body elements and what is the result. And there is this probe. Consider for example, there is some type of reaction occurring in our body and we have to know what is that reaction. So we conduct similar experiment with those reactants outside in lab under conditions, specific conditions. Since in outside we have air and everything, so they react more. But so under the specific conditions, of course, we can conduct experiments. From that, we will compare, conclude to a solution that this may be the reaction happen. So this will be also studied in bioinorganic chemistry, and we'll confirm it using various characterization methods. Before going into metalloporphyrin, we must be knowing the porphyrin molecule. So rest of the things are derivatives of porphyrin molecule. So this porphyrin molecule has this pyrrole group. See, we can see here 4 pyrrole groups are present. See here in this nitrogen and this nitrogen has a hydrogen group, but this both doesn't have any. But as we can see here, all the nitrogens have 3 bonds and a lone pair according to octet rule. Only then they are stable. And then we can also notice that there is conjugation in the perimeter, that is the boundary. So these are all the some things we have to notice so that we can draw the structure very easily. And then see here, there is a bridge between every pyrrole group. There is like 4 bridges. I have mentioned alpha, beta, gamma and delta. We can also number accordingly so that we'll get 5, 10, 15, 20. At these positions we have this methylidine group. So this structure is very important. So start drawing, and it is very easy if you know what is the number of bonds for nitrogen and how this conjugation is placed everywhere and similar information. So porphyrin. Porphyrin is nothing but the derivative of porphyrin molecule. This porphyrin will have substituents in their perimeter. I have not mentioned anything here, but it contains substituents, only then it is called a porphyrin. When this porphyrin donates 2 hydrogen, this I have mentioned for your convenience. If it is donating its H plus ions, then the electrons in this bond will shift to this nitrogen in both the cases, so that we will get a dianion. Similarly, if it is accepting 2 H plus ions, consider H plus ions means they won't have electrons. So of course the bond should be formed by the electrons present in the nitrogen. So this nitrogen will donate its electrons and the bond is formed. Now they have 2 positive charge. So what we conclude is donating 2 protons will give a dianion and accepting 2 protons will give a dication. So this is the cavity where it can attach any metal, especially first transition metals. In a book I have seen that it accepts Ni2 plus very well than others. Then metalloporphyrin. So metalloporphyrin means metal is present in the porphyrin structure. That's it. So the metal is attached to the 4 nitrogen groups and it forms a square planar geometry, as you can see here. So notice here that this nitrogen and this nitrogen has 3 bonds, but this 2 nitrogen doesn't have 3 bonds, it is having 4 bonds. So this is not actually a bond. In most of the books they have mentioned it in dotted lines. Okay, this is maybe due to electrostatic attraction, but this too is of course a bond. How is it formed means, like I have mentioned here, it will donate the 2H plus outside, so that there will be two anion, that is dianion in the structure, and this dianion will form this bond with the metal. Then there is something to notice that it can also attach something above and below the square planar structure. So if it is attaching something above and below, then we will have a octahedral geometry. Here we can see that in oxyhemoglobin it has attached dioxygen and histidine group, and we have an octahedral geometry as I have mentioned. So then we have to know that this is very rigid. Why is it rigid? Means because there is conjugation around the perimeter. This is not only the example for this, there is also some other examples like myoglobin, cytochrome, chlorophyll. These are also something that have this porphyrin with various ligands in axial sites. Heme group. So why do we have to learn heme group? See, generally there are some proteins we'll classify like heme protein, non-heme protein. So what is heme? So heme group, it may be also called Fe protoporphyrin 9. So basically we can tell like it is a metalloporphyrin where iron is the metal. That's it. So this is the structure. So this looks a little bit complicated, but this is fine, right? We can draw this. So this is, we have to practice to draw this. Here we can see there are around 8 substituency. 1, 2, 3, 4, 5, 6, 7, 8. So 8 substituents. And at the 9th time, the cycle is starting again, as you can see. So that's why it is named 9. So why is this proto? Proto means this is protonated, COH, propionic acid. That's why we are calling it this protoporphyrin. Then coming to the oxidation state, it has +2. How do we calculate it? Usually we will calculate oxidation state based on the charge of the ligands. What is the charge of this ligand? Here we can see that already I have mentioned how this bond is formed due to the dianion after donation of 2H+. So of course the ligand will have a charge of -2. See, from here this will donate its 2H+, and so it will have dianion. Then these electrons will form a bond with our metal. Thus it will be having +2 oxidation state. See, this is the charge of the ligand, -2. X is the oxidation state of Fe is equal to 0 because the overall charge is outside, they haven't mentioned, so it is 0. Then X will be 2. So Fe configuration is argon 3d6 for S2, and Fe2 plus will have a 3d6 configuration. So that's it for today. This is a very basic thing, I know, but it is very important to understand it very deeply, so that later on whatever you learn, it will be easy. So my reference books are Lippard and Burke, Ajay Kumar. Everything is available online. Of course you can take it from anywhere, especially there is a website called Z Library. From there you can download it. Then thank you for supporting me by watching this video. If you have any doubts, of course you can tell in comments. I would love to answer it. I'll refer anything and I'll try to answer you. Thank you for watching.